Have you ever wondered how scientists separate different substances? You might be surprised to learn about a powerful technique called chromatography. It's essential in many fields, from medicine to environmental science. Today, we'll explore how chromatography works and why it matters. Chromatography is a method for separating mixtures into their components. It allows scientists to analyze substances precisely. This technique plays a critical role in identifying unknown chemicals in a mixture. Let's break down how it works, step by step. First, we need a mixture to analyze. This can be anything from plant extracts to environmental samples. Once we have our mixture, we prepare it for chromatography. Next, we choose a stationary phase. This is the material that remains fixed in place during the process. Common examples include paper or silica gel. The stationary phase is critical for separating the components. After selecting our stationary phase, we then introduce the mobile phase. The mobile phase is a solvent or gas that travels through the stationary phase. It carries the mixture with it, allowing separation to occur. Now, let's visualize this. Imagine a simple paper chromatography setup. We apply a small drop of our mixture on a line of the paper. As the paper absorbs the solvent, it travels upwards. The components of the mixture move at different speeds, creating distinct spots. This separation occurs because of the differences in interactions between the components and the stationary phase. Some substances are attracted more to the stationary phase, while others are drawn to the mobile phase. This creates a separation of colors or substances. To help illustrate this, think of food coloring. If you mix different colors in water, chromatography will separate them. You can see how each color travels at varying rates along the paper. Once the components are separated, scientists analyze the results. Each spot corresponds to a substance in the mixture. By measuring the distance traveled by each component, scientists can identify them. In summary, chromatography is a powerful separation technique. It allows scientists to identify substances effectively. By combining a stationary phase with a mobile phase, mixtures can be analyzed and understood. If you're interested in learning more about chromatography, check out our other videos. Don't forget to subscribe for more scientific explorations. Thank you for watching.